There's lots of different chemical compounds dissolved within our blood plasma. Some of them are going to be nitrogenous chemical compounds. When we nitrogenous chemical compounds, these are chemical compounds that have a nitrogen atom in them. In particular, this nitrogen atom is going to be located most of the time within an amino acid. By far, the most common nitrogenous waste product that's present in our blood plasma is urea. This urea gets its name because this is a chemical that was actually isolated from urine, hence its namesake, and now we call it urea. Urea is also a precursor to uric acid. So if you ever wondered why dirty, sweaty gym socks smell so bad, it's the urea that made it from the blood plasma to your sweat. And then in your sweat, that urea turns into uric acid, and from uric acid to ammonia, which is another nitrogenous chemical compound. We also have our generic nutrients, so our carbs, our lipids, our proteins. We're going to have lots of gases, in particular dissolved oxygen gas, a small amount of dissolved nitrogen gas, and a very incredibly small amount of dissolved carbon dioxide gas. We actually don't carbonate our blood that much, thankfully. And then finally, we have electrolytes. Think all the way back to last semester. An electrolyte is a chemical compound that conducts electricity when dissolved in aqueous solution. More practically speaking, it's an ion, more generically speaking. Now, as we're looking at our blood plasma, a big thing that our blood plasma is going to influence is the viscosity of our blood. When we think of viscosity, I want you to think how liquidy or how runny. It's also going to influence osmolarity of our blood. We'll talk about osmolarity on the next slide. Now, when we think of viscosity for our blood, because our blood has water plus a bunch of formed elements and solid chemicals mixed in with it, it's pretty safe to assume the blood is more viscous than our water. It's thicker. It doesn't flow as smooth as water. Measuring viscosity is water. And water is defined as having a viscosity of 1. So when we look at our blood, whole blood, depending on your water concentration, depending on how many red blood cells you have, could be 4.5 to 5.5 times more viscous than water. Even the plasma, which is overwhelmingly water, is still going to be more viscous than pure water because we have those dissolved ions, those dissolved nutrients, and in particular, we have a lot of dissolved proteins, those globulins, the albumins, and the fibrinogens present within our blood plasma. Now, we mentioned osmolarity very briefly last week after I bombarded you with syllabus policies. And when we think of osmolarity, I want you to think of a quantification of the osmotic potential of a liquid. Osmolarity is a way of using a number to measure the tendency of a liquid to pull water into itself, or the tendency of a solution to pull water in. If our blood has a higher osmolarity, it's more likely to suck water into its blood plasma and decrease its viscosity. When you're dehydrated, you generally speaking are going to lose water from your blood plasma, and the blood viscosity and the blood osmolarity will both go up. We're not going to spend time doing mathematical calculations of osmolarity. For those of you who really like chemistry, you take the molarity of your dissolved solutes and then divide those or multiply the dissolved solute molarity by the number of ions that dissolved solute forms, and that gives you the osmolarity. But I'm never going to ask you to do that calculation. Now, if our blood osmolarity is too low, let's say we don't have enough albumin in our blood, that protein we talked about last week. If we don't have enough albumin in our blood, our osmolarity drops down, and then our blood has a tendency to lose water from the blood plasma. We have this ability, we have a hard time maintaining the correct viscosity of our bloodstream. Now, when we look at our osmolarity, because it's going to very tightly regulate our water balance, we have multiple organ systems that help to regulate blood osmolarity and blood viscosity. Moving on, let's talk about how we make blood. This is a number that we just need to pause and talk about. The number of red blood cells we make every day, the number of erythrocytes we make on a daily basis is 100 to 200 billion red blood cells every single day. And within those red blood cells, they're pumping out several more billion hemoglobin molecules per red blood cell, within our red blood cells every single day. So if we think back to mitosis that we talked about last semester and protein synthesis, we focused in on one ribosome. We focused in on one dividing cell line. Take that number, that process, and multiply it by several hundred billion, and we do that every single day. It's very quickly going to become something that's overwhelmingly complicated, and we just do it automatically. I think it's really cool. The old school name for making blood is hemogenesis. Hem referring to blood. Genesis is a root word that means create, or to create in the Latin. Think of like that book of the Bible that has the story of creation. The same root for Genesis there is when we had hemogenesis. The new school term that you'll see in the newer textbooks as opposed to the older literature is poiesis. And it's the same meaning, just a different root word. It means to create. So when we have hemopoiesis, that's making blood. And hemopoiesis is an umbrella term. Subcategories under the umbrella of hemopoiesis include erythropoiesis, making red blood cells, leukopoiesis, making white blood cells, and thrombopoiesis, making platelets or thrombocytes. Now, depending on where you are developmentally as an individual, you're going to have hemopoiesis taking place within different tissues in different parts of your body. When we were baking in the oven, so to speak, when we were a fetus in utero, we had a yolk sac. And the yolk sac is where we had our first red blood cells, or our first blood cells really come from. So when we think of that yolk sac, I want you to think of fetal origins of red blood cells. And then as we grow, we're born, we keep growing, we're going to have more red blood cells being produced. Organs at play include the liver and the spleen that help to make more red blood cells. When we talked about the primary skeletons, it's worth writing in the margin that within adults, most red blood cells are going to be produced within the red bone marrow of flat bones. So we would be remiss if we didn't emphasize the red bone marrow. Now within our red bone marrow, we have multipotent stem cells. Let's pause for a moment and just review briefly what we talked about with stem cells. There are different kinds of stem cells in our bodies. Depending on how flexible that stem cell is, maybe it can turn into a couple different kinds of cells, or maybe it can turn into many, many different kinds of cells. The most flexible kind of stem cell in our body is the totipotent stem cell. They can turn into human tissues that are part of your body, or they can turn into a placenta, which is not part of your body. We don't walk around with a placenta hanging off of our navel. Then we have pluripotent stem cells. Pluripotent stem cells can form any cell line within a human body. And we have lots of pluripotent stem cells when we're in utero. Another name for pluripotent stem cells is embryonic stem cells. And then finally, we have multipotent stem cells. Another name for multipotent stem cells, and you can write this in the margin, is adult stem cells. When somebody talks about an adult stem cell, they're talking about a multipotent stem cell. A stem cell that can turn into a couple different kinds of cells, but doesn't have the kind of flexibility that a pluripotent or a totipotent stem cell would have. So when we look at a hemocytoblast, this is a kind of multipotent stem cell that is going to make blood. Heme is a word that means blood. So I'll go ahead and circle that for you. Let's go with green. So hemo meaning blood, cyto referring to cell, blast means to build. It's a blood building cell. A hemocytoblast forms blood cells. And that's the old school name. The new school name that's being phased in is the hemopoietic stem cell. Same thing. Now, as we look at these multipotent stem cells, they can be further differentiated into a colony-forming unit. these colony-forming units are going to make a very specific kind of blood cell or formed element within our blood. Now we are going to have hemopoiesis, or blood generation, the creation of blood, taking place in two primary tissues as an adult. We're going to have myeloid hemopoiesis in our red bone marrow, and then we're going to have lymphoid hemopoiesis that takes place within our lymphatic organs. When we think of myeloid hemopoiesis, I want you to primarily focus on erythrocytes. When we think of lymphoid hemopoiesis, I want you to primarily focus on leukocytes, or white blood cells. So here's our technology tree, or the tree that shows how our hemocytoblasts can differentiate. We have the parent multipotent stem cell, and then that cell can differentiate into precursor cells, or colony-forming units. And depending on the precursor that it differentiates into, some of those precursors will turn into multiple cell types. Other precursors will only turn into a single cell type, depending on the precursor cell that multipotent stem cell differentiates. For exam purposes, I am not going to ask you to regenerate this figure. What I want you to primarily focus on, because I know you're always thinking, well, how's he going to ask a question about this? I would ask you a question like, what is the stem cell that generates a erythrocyte? And I would want you to say a hemocytoblast, or a hemopoietic stem cell. I don't want you to take a lot of time to memorize these intermediate cell types, like the prochromatic erythroblast or the progranulocyte. We're not going to focus on those in particular. So don't spend mental energy focusing on those intermediate cell types. I'm really interested in the beginning and the ending. Speaking of the ending, the cell that's produced the most at the end is our red blood cell, or our erythrocytes. And when we look at erythrocytes, they have two primary functions, but really to me there's one primary function. The single biggest function of a erythrocyte, in my mind, is to carry oxygen gas. Our red blood cells, our erythrocytes, carry oxygen gas. They're suitcases filled with oxygen, or you could think of them as a wheelbarrow filled with oxygen. They pick up oxygen in our lungs, and then they go to our tissues, dump the load, and then they go back to our lungs and pick up more oxygen, and they just repeat the process over and over and over again for about 120 days. Another very important function of our erythrocytes is that they help us to transport carbon dioxide. And I'll spend some more time on this nuance of carbon dioxide, but I'm going to say it now because this is a common student misconception. Most of the carbon dioxide in our bloodstream is not transported as carbon dioxide gas. Most carbon dioxide in our bloodstream is turned into bicarbonate ions. And this is a good thing, because if we had lots and lots of carbon dioxide gas dissolved in the bloodstream, whenever somebody flicked you, your blood would fizz just like a can of pop. And having bubbles in your bloodstream is never a good thing. Our red blood cells take that carbon dioxide, put it through some chemical reactions, and turn it into some ions or some electrolytes, and then we transport it as a dissolved ion, not a dissolved gas. Now, when we look at our red blood cells, there's some variation in how big they are. We're going to talk about an idealized average erythrocyte. An average erythrocyte is about 7.5 microns in diameter, too small to see with the naked eye. You can see them with the 400x microscope, visible light microscopes. And in lab this week, we're also whipping out the oil and using the 1000x magnification. So we can really see some of those differences in the leukocytes. Another key characteristic of our red blood cells is that they have a biconcave shape. In other words, there's two big indentations, biconcave. Or they look kind of like a jelly donut with an indentation in the middle. And as we're looking at these red blood cells, the reason they have the biconcave shape is because they've lost most of their organelles during their developmental process. A red blood cell primarily is going to transport oxygen gas inside of itself. And in order to do that, it doesn't waste any energy with the nucleus. It doesn't waste energy with ribosomes. It focuses almost exclusively on just carrying these gases, this oxygen gas around. So because our red blood cells have lost almost all of their organelles, the cytoplasm shrinks in or invaginates on itself and gives it the biconcave shape. Now, think back to when we talked about cellular respiration last semester. We had glycolysis leading to the citric acid cycle, leading to the electron transport chain. And of those three steps, the citric acid cycle and the electron transport chain take place in mitochondria. So we can't have full-fledged generation of ATP, but glycolysis takes place within the cytoplasm of the cell. And because red blood cells have a cytoplasm, we can have glycolysis taking place so they can make just a little tiny bit of ATP through anaerobic respiration, respiration without any oxygen gas being used up in the process. And if you think about it, this is a good thing. You don't want your red blood cells to use up the oxygen that they're trying to deliver. It's kind of like the pizza delivery boy eating most of the pizza before they get to your house. That'd be a lousy delivery boy. And in the same way, a red blood cell should not consume the chemical product that it's trying to deliver to the other tissues in your body. There's a very important reason why we don't have any mitochondria within our red blood cells. Now, when we think of blood typing, our blood type is going to be determined by the surface of the cell. Last semester, we talked about how there's glycoproteins, glycolipids. There's all these different moieties, or these different chemicals that label the surface of our cells. These chemicals on the surface of the cell help our body determine if it's a blood cell or a cell that belongs in a muscle tissue, or maybe it's a bacterial cell, or maybe it's a cell that's infected by a virus. These surface markers are what we use to identify the cells in our bodies. And within our blood types, we think of like A, B, O, and then the positive and negative for our blood types. The surface proteins and the surface carbohydrates help to determine what our blood types are. We also are going to have proteins on the inside of our red blood cells, and particularly spectrin and actin. These are the two primary proteins that make up the cytoskeleton of our erythrocytes. We talked about actin last semester. Spectrin we did not talk about last semester. That's a new cytoskeletal protein that I'm introducing right now. Spectrin and actin are the two cytoskeletal proteins of erythrocytes that allow our erythrocytes to stretch, bend, and squeeze. I'm going to emphasize bending. Our erythrocytes need to be flexible. Blood capillaries are the smallest blood vessels in our bodies, and some of those blood capillaries are going to have a diameter of as low as 10 microns for that inner diameter. So if we have a red blood cell that's a little bit larger than average, maybe that red blood cell it's going to have a very hard time squeezing through a 10 micron blood capillary, and it's going to need to be flexible to get through that capillary. We're going to spend a lot of time camping on this slide. There's some very key concepts on this slide. I can't emphasize the surface area to volume ratio enough when we're looking at our erythrocytes. Because our erythrocytes have that biconcave shape, they have a very high surface area to volume ratio. And this very high surface area to volume ratio allows for gases like oxygen and carbon dioxide to rapidly diffuse across the membrane, the cell membrane of that erythrocyte. There is a classic high school biology lab. I hope every one of you had the opportunity to do this, where you have agar cubes filled with pH indicator. And you have the tiny agar cube and the medium agar cube and the really fat big agar cube. And you throw them into a sodium hydroxide solution and then watch and calculate the rates of diffusion. And you find that the agar cubes that have the highest surface area to volume ratio can have the chemical compound diffuse across it the quickest compared to the agar cubes that are ginormous. By having this very high surface area to volume ratio, we optimize the red blood cells to be the best possible transporters of oxygen gas. The key unifying idea in this class is that the function dictates form, or form follows function. The shape of the red blood cell makes it really good at transporting oxygen gas across its cell membrane and then transporting that oxygen gas around our bodies. Inside of our red blood cells, the single most common protein is going to be hemoglobin. And because we're going to talk about hemoglobin so much in this unit, we're going to abbreviate hemoglobin with a capital H and lowercase b. As we're looking at that hemoglobin, we have about a little bit over 250 million hemoglobin molecules per red blood cell. So if we're making 200 to 300 red blood cells per day, and each red blood cell is having 250 million hemoglobin molecules, just imagine all the proteins those ribosomes are pumping out. It's very complicated when you look at the big picture. Now, in our erythrocytes, there's a protein that's much rarer. There's not as much of this protein compared to the hemoglobin. It's called carbonic anhydrase. And you may notice it ends with an ASE. That ASE suffix at the end is the nomenclature of an enzyme. Because it ends with an ASE, it's going to be an enzyme. What does this enzyme do? It is going to remove hydrogen from carbonic acid. So an means to remove. What does it remove? A hydrogen from a carbonic acid. This is the enzyme that catalyzes the chemical reaction where carbon dioxide will come into a red blood cell, and that carbon dioxide will ultimately be turned into a bicarbonate ion that's then dissolved in our bloodstream. This is very, very important for allowing us to move carbon dioxide in our bloodstream, but at the same time not carbonate in our bloodstream. And you can imagine, as we're going through this process of adding and subtracting hydrogens from the chemical compound, we're going to alter the pH of the solution, or the hydrogen ion concentration of our bloodstream. So in addition to helping us transport carbon dioxide, carbonic anhydrase is also a very key protein for regulating our blood pH and helping us maintain homeostasis with regards to our blood pH. So, pull out your cell phones. We have a quick question for us. Following statements about red blood cells is false. We have A, large amounts of, think of the carbon dioxide gas. I should have put gas at the end of that option. Red blood cells transport large amounts of oxygen, gas. Red blood cells have large surface area relative to volume in order to speed diffusion. Or red blood cells are rigid and will not flex in capillaries. Now that I'm reading this out loud, there's 2 correct answers to this one. I should have drank more coffee. Red blood cells do not transport large amounts of carbon dioxide. They take the carbon dioxide in and turn it into carbonic acid and then the bicarbonate ion. Red blood cells do transport large amounts of oxygen. They do have a large surface area relative to volume. They do not have a rigid composure. So I screwed up here. I should have said red blood cells are flexible so that they can move through capillaries. So the 2 correct answers are A or D for this option, for this question. Not just A. But of these answers, B was absolutely the more false. It's more straightforward. So I understand why we selected D. But A is also incorrect. Red blood cells do not transport large amounts of carbon dioxide. Let's talk about hemoglobin. Ruby. Really good question. So Ruby asked, How can hydrogen be removed from carbon dioxide if carbon dioxide is one carbon and two oxygens and doesn't have hydrogen on it? I'm glad you're thinking about it. Can I insert a blank slide? I really don't want to write on the board. Oh, I know what I can do. I will go to Create, and I just need to, there we go, whiteboard sketch. So I'll present this slide for us. So carbon dioxide is a, for the record, you do not need to know this right now. I'm answering Ruby's question. Carbon dioxide doesn't have a hydrogen, it's CO2, but there's lots of water in the bloodstream, and that water has hydrogen on it. And then we can have some double reaction arrows and have H2CO3, which is the carbonic anhydrase, and then we can have another double reaction arrow and have the H positive, the hydrogen ion, plus a bicarbonate ion, an HCO3 with a -1 charge. Carbonic anhydrase is going to remove the hydrogen ion from this intermediate in the middle, the carbonic acid, not from the carbon dioxide. Where was I? Hemoglobin. There we go. Oh, it's not presented yet. So, when we think of hemoglobin, I want you to think of the most common protein inside of the red blood cell. This is the protein that's going to move the oxygen molecules. This is the little wheelbarrow, so to speak, that you fill up with oxygen and then you dump out the oxygen. And there's two variations of hemoglobin. We have adult hemoglobin and fetal hemoglobin. And they have two different affinities. When we think of development in utero, the fetus doesn't have lungs. The fetus can't open its mouth and breathe air in and out, oxygen gas. The fetus has to get all of its oxygen from its mother's bloodstream. And fetal hemoglobin has a higher affinity for oxygen. It has the ability to pull oxygen off of adult hemoglobin to oxygenate the fetus's bloodstream. Now, as we look at hemoglobin in general, hemoglobin is a 4-subunit protein. Adult hemoglobin will have 2 alphas and 2 betas for the subunits. The betas are the kind of olive-colored subunits on the screen, and then the alphas are the purple subunits on the screen. And when we look at fetal hemoglobin, it has a different chemical composition from adult hemoglobin. That different composition gives it a different structure. The different structure makes it more likely to bind to oxygen than in the adult hemoglobin. Within the hemoglobin, each globular subunit has a heme molecule. The heme molecule is shown on the bottom of the screen right here. And as we look at this heme molecule, we have the all-important iron atom right in the center. That's where the action is at. When we add an oxygen to a hemoglobin, that oxygen gas molecule is physically attaching to the iron atom in the heme subunit of the hemoglobin. In other words, when we oxygenate our bloodstream, it's not an exaggeration to say we rust the blood because we take oxygen and attach it to iron. We make iron oxide or rust, just a little tiny bit. And the reason our blood is, oxygenated blood is bright red is because iron oxide is a very brightly colored inorganic chemical compound. Now, as we look at this, we need to do some simple math, counting, so to speak. We have 4 globular subunits in a hemoglobin. We have, in each globular subunit, a heme molecule, and in each heme, we have 1 iron. So if you backtrack the math, each hemoglobin can hold 4 oxygen molecules, 4 oxygens per hemoglobin, because we ultimately have 4 attachment points. This is not a plant physiology class, and I'm absolutely not going to test you on this, but if you are interested in plant phys, you can make a chlorophyll pigment by taking the iron and replacing it with a magnesium atom. We're one atom away from green blood. So, as we're looking at our hemoglobin, we can measure the amount of red blood cells and indirectly the amount of hemoglobin in our blood by taking our blood and centrifuging our blood. Once we've centrifuged our blood, we've created what's known as a hematocrit. And the hematocrit is a way of just measuring the amount of formed elements relative to the amount of plasma in the bloodstream. Now, as we look at males, generally speaking, males don't have menstrual cycles. Therefore, they're going to have more red blood cells in their bloodstream. Women, particularly of a reproductive age, are going to have menstrual cycles, so they'll lose more red blood cells on a monthly basis and have a lower percentage of red blood cells in their hematocrits. Now, when we think of the hemoglobin concentration, the concentration of hemoglobin is going to vary a little bit from individual to individual. And when we look at the gender differences, generally speaking, males have a slightly higher concentration of hemoglobin in their blood because males have just a little bit more blood cells in the bloodstream compared to females. When we look at counting our red blood cells, the concentration for males is about 4 to 6 million red blood cells per microliter, whereas for females, 4 to 5 million red blood cells per microliter. By far, the biggest factor that lowers red blood cell counts for females is going to be the menstrual cycle, the regular loss of blood. But there are other factors as well that are going to cause values to be lower in females. In particular, males are going to have more sex hormones, or testosterone in particular. Yes, males also have a fair amount of estrogen in their bodies as well, but it's the additional testosterone that really upregulates the red blood cell production. We also find that as we look at our hematocrits, we're going to have a relationship between percentage body fat and hematocrit production. If you have more androgens, if you have more testosterone in your blood, generally speaking, you have more skeletal muscle and less fat in your body. So we have this inverse relationship. As the percentage of fat in your body goes up, the concentration of red blood cells in your blood, because there's a correlation between fat and androgen concentration in our bodies. Let's talk about erythropoiesis. When we think of erythropoiesis, I want you to think of making red blood cells. The old school name is erythrogenesis. And as we're looking at erythropoiesis, we start with a multipotent stem cell. That multipotent stem cell will then form some precursor cells, notably the collagen-forming unit, and then eventually get to the mature erythrocyte. Now, as the erythrocyte, as the red blood cell is developing, it has proteins in it. You know, it has lots of hemoglobin, it has lots of carbonic anhydrase. These proteins had to be made by ribosomes. There had to be some genetic code, some RNA to program the ribosome. So at one point, there was a nucleus. The precursor cells have a nucleus, have quite a few organelles. But as the erythrocyte is developing, these organelles self-destruct, they disintegrate, and eventually you're left with a much smaller cell that has that biconcave shape. Now, when we think of this on a per second basis, we make about 1 million red blood cells every second we're alive. Pumping out 1 million red blood cells per second. It's kind of crazy when you think about it. These red blood cells last for about 120 days is the average lifespan. That's about 1/3 of a year. So when we think about how our blood is replaced, we replace all of the blood in our body about 3 times a year. We completely recycle and replace every red blood cell in our bodies. Well, as we're making our red blood cells, it takes 3 to 5 days to make our red blood cells. There are some factors that can influence the production of red blood cells. If you have enough iron in your diet, It's really easy to make hemoglobin, and the red blood cell production rate is going to be upregulated. If you don't have enough iron in your diet, it's kind of hard to make more red blood cells. And then there's other factors as well. One of the most important factors is a chemical hormone known as erythropoietin. Erythropoietin, or EPO, is a hormone that stimulates the production of red blood cells. Depending on the process, occasionally there's a rush order put in for our red blood cells. And instead of having mature red blood cells in the bloodstream, every once in a while you'll see a homeostatic imbalance or a disease state where an individual will have a precursor red blood cell, a nucleated red blood cell, dumped into the bloodstream. You should never see that in a healthy individual. If there are nucleated erythrocytes, that means that that individual is under a severe amount of stress and is suffering from a lot of oxygen deprivation. I'd mentioned this just a little bit ago. Iron is the key nutritional requirement for red blood cell production. It's generally speaking going to be the single biggest limiting factor. We lose a lot of iron every single day. We need to tightly regulate iron. Iron is not only a limiting factor for red blood cell production, iron is also a limiting factor for bacterial growth in our bodies, particularly in our blood plasma. So we have to very tightly control the amount of iron in our bodies. We want lots of iron for our red blood cells, but we don't want a lot of iron for sepsis. We don't want bacteria growing out of control in the bloodstream. So there's this fine line, kind of a razor's edge that we balance on. Now, as we look at males versus females, we have differences in the loss of daily iron. Men typically lose about 1 microgram of iron. Females lose about 2-ish micrograms of iron per day. So one of the biggest differences you see between male multivitamins and female multivitamins is the presence of iron in that multivitamin. A good woman's multivitamin will have extra iron in it to help supplement the iron loss. I right now am taking a postmenopausal woman's multivitamin because my wife bought it by accident. And that multivitamin doesn't have the extra iron in it, so it's basically an adult male multivitamin. Menstrual cycles. Overwhelmingly menstrual cycles. So Ruby asked, why do females tend to lose more iron? And the answer is they tend to have a menstrual cycle. We find that we have a really hard time absorbing iron in our bodies. Most of the iron that we consume is not going to be absorbed in our bodies. Some breakfast cereals overcompensate by including way too much extra iron in them. A classic freshman science lab activity is taking Wheaties, grinding it up in a plastic bag and dropping a magnet in the plastic bag, and then pulling out the chunks of iron flakes from the breakfast cereal. They really upregulate the amount of extra iron they put in wheat. To help control the amount of extra iron that we have available for bacteria, we have a protein that helps regulate free iron concentrations called ferritin. Ferritin is going to be produced by our liver and will store iron in such a way that we can use it to make red blood cells, but that it's hard for bacteria to access. So we're going to camp on this slide for a bit as we're looking at iron metabolism and how we're processing our iron. First, we eat the iron. And that iron goes into our stomach, and it goes from a +3 oxidation state to a +2 oxidation state. Well, it's in our stomach. So from Fe +3 to Fe +2. And then while it's in our stomach, there's a chemical compound or protein known as gastroferritin that will take that iron from our stomach and help transport it to the small intestine where it's going to be absorbed. After being absorbed in the small intestine, it goes into the blood to another protein called transferrin. And if you look at the name of these proteins, the root word for iron is in the name of all of these proteins. Ferrin for gastroferrin, transferferrin, aptoferrin, or ferritin. These are all proteins that deal with the ferrous element, or with iron. Ferrous is the Latin word for iron. So transferrin brings the iron to the liver. Within the liver, we're going to store the iron on an aptoferritin that's been converted to a ferritin. And then once we need that iron, we can dump it from the liver into the bloodstream. Let's talk about regulating our red blood cell concentration in our body. The process of making red blood cells is known as erythropoiesis. That's the new school name. And this is a classic negative feedback loop. So if we have too much red blood cells, we generally speaking stop making red blood cells. We don't want to have too many. If we don't have enough red blood cells, we start to make more of them. We keep going towards the homeostatic balance point where we're making our red blood cells. Now, if we think of red blood cells as the oxygen carriers in our bodies, if we have enough red blood cells, we're going to have enough oxygen in our bloodstream. But if we don't have enough red blood cells, we don't have enough oxygen, blood oxygen levels go down. And an organ in our body that's very ATP dependent, that uses up about 20% of our ATP, is our kidneys. And our kidneys are very sensitive to fluctuations in blood oxygen concentration. And as a kidney senses a drop or a decline within our blood oxygen levels, a decrease in blood oxygen will stimulate the kidneys to make a hormone called erythropoietin. And then erythropoietin, because it's a hormone, is going to be dumped into the bloodstream. It'll be transported through the bloodstream to go to the red bone marrow of our flat bones. Once the erythropoietin is in our red bone marrow, it's going to stimulate erythropoiesis. And then as we have erythropoiesis stimulated, we have more red blood cells and blood oxygen levels go back up again. And there's a 3 to 5 day delay on this process. So when we think of making red blood cells, the single biggest factor that stimulates red blood cell production is going to be low blood oxygen levels. Now, if you're stuck at one elevation and you don't travel a lot, and you have low blood oxygen levels for an extended period of time, that's usually going to indicate a disease state or some kind of homeostatic imbalance. High-performance athletes, for years and years now, traveled to high altitudes or high elevations, like Denver, Colorado, where there's lower oxygen concentrations in the atmosphere, and that will artificially lower blood oxygen levels so that they make just a little bit more red blood cells, and then after acclimating to those low oxygen, high altitude environments, they go back down to sea level and compete at the Olympic Games and do amazing things. Other things that can cause erythropoiesis to be stimulated include exercise. If you exercise more, you will have more red blood cells. This goes hand in hand with that inverse relationship that if you have more body fat, you have fewer red blood cells. If you exercise more, generally speaking, you will lose some body fat and have more red blood cells. And then also, during some disease states of the lung, like emphysema, where we have a hard time of putting oxygen into our bloodstream with the lungs, when we have those lung or pulmonary diseases, that can stimulate erythropoiesis as well, because that lowers the blood oxygen level. Now, when we get rid of our red blood cells, because they don't last forever, they're going to rupture, they lyse, we have hemolysis, the destruction of a red blood cell, and we need to process that iron. And most of what we're going to focus on is how that iron is processed. We can't have extra iron floating around in the bloodstream. That's bad for us. So as we're processing our red blood cells, generally speaking, they're going to go through hemolysis or rupture in the organ of the spleen and the liver to a lesser degree. And as these organs are processing our red blood cells, we're going to take the leftovers and they'll be digested by a white blood cell known as a macrophage, or the big eater. And then within those organs, we're going to remove the heme from the globular component. The heme is the molecule that has the iron in it, The globin is the protein component of hemoglobin. We take that protein component, the globin, and we just chop it up into individual amino acids and then recycle and reuse those amino acids, kind of like taking a Lego set apart and building something different out of it. The iron, on the other hand, is going to be stripped from the heme subunit. And that iron and heme pigment are going to be processed to biliverdin. Biliverdin is present in high concentrations in bile. Bile is a liquid that's present in our digestive tract, and the reason bile is green and the gallbladder is green on most lab models is because of the high concentrations of biliverdin present in the bile. But within our body, particularly within our digestive tract, we're going to have lots of bacteria that can convert the biliverdin into bilirubin. Bilirubin is one you've probably heard of. Individuals that have jaundice or yellow skin have too much bilirubin in their bodies. Particularly, if you get newborn infants that are born prematurely, they'll be subject to elevated bilirubin levels, or individuals with liver failure, adults with liver failure, maybe they have hepatitis, for instance, they'll have yellow eyes instead of white eyes because they have elevated bilirubin levels in their bloodstream. That bilirubin goes to the bloodstream. From the bloodstream, the kidneys will remove a lot of the bilirubin and we urinate it out. That's what makes our urine yellow. Our liver, though, is going to take most of that bilirubin and dump it into the bile and then release it into the intestine. From our intestinal tract, we can take that bilirubin and process it into urochrome or urobilirubin, which is what gives us the brown coloration to human feces. So when we think of daily loss of iron, urination and defecation are two very big sources of iron loss in our body. It's worth mentioning that individuals that have elevated bilirubin levels are going to have elevated urobilirubin levels in their feces. Individuals that have jaundice oftentimes will have black feces because the urobilirubin concentrations are so abnormally high. Instead of being a brown color, there'll be a black tarry coloration to the feces. When you become parents, you get very interested in things like diapers and stuff like that. So when we think of a erythrocyte is going to start out with some iron in our intestinal tract. We absorb the iron and then have erythropoiesis in our red bone marrow. That erythrocyte spends about 120 days or about 4 months moving around in our bodies, delivering oxygen, transporting gases, doing its thing. And then after about 4 months, the red blood cell is going to be disintegrated in the spleen and or liver. The globular component is chopped up into amino acids. The heme component is going to have the iron removed from it, and then we are going to make bile and bilirubin and urobilirubin from those leftovers. Sometimes we have too many blood cells in our body. The condition of having too many red blood cells is known as polycythemia. And there's two variations of polycythemia. There's intentional and unintentional. When we think of unintentional polycythemia, so we have too many red blood cells in our bodies, and we aren't intentionally trying to do that, that's going to be referred to as primary polycythemia, also known as polycythemia vera. This is the disease. I'm going to put a big D there for disease because that's not intentional. It's a loss of homeostasis, and it's outside of the person's control. The most common cause of primary polycythemia is going to be cancer of the colony-forming units within the red bone marrow. So if those colony-forming units are reproducing out of control, we're going to have out-of-control red blood cell production in our bodies. We can also have secondary polycythemia. Secondary polycythemia, generally speaking, is not going to be a disease state in a lot of individuals. It can be caused by a disease state. Maybe there's emphysema. That's a disease state that can cause secondary polycythemia. But most instances of secondary polycythemia are going to be from exercising a lot, or maybe you're hanging out at a high altitude or a low oxygen environment, or you're temporarily dehydrated. If you want to artificially inflate your hematocrit, you should drink a lot of coffee right before you go to donate blood. It will definitely cause them to raise some eyebrows when they test your blood. I'm going to date myself here, but in the mid to early 2000s, Lance Armstrong was one of the most famous professional bicyclists in the world. He was known for winning the Tour de France multiple times. He had found a way to game the system in that he would inject himself with erythropoietin several weeks before competition. And by injecting himself with erythropoietin, he would artificially increase his red blood cell concentration and have an increased, in theory, increased blood oxygen carrying capacity in his bloodstream. It was since figured out what he was doing. His titles were stripped from him. But I linked here a really interesting research article that was published in 2017 that found that the increase of blood oxygen carrying in the bloodstream is counteracted by the increased viscosity of the blood. If you have too many red blood cells in your bloodstream, it's harder for your blood to move. You have less water, and it's harder to transport oxygen from point A to point B, even if the carrying capacity of the bloodstream has been increased. So what I found particularly ironic about this study was that injecting yourself with erythropoietin has no net effect on athletic performance. Too bad nobody told Lance that. So when we think of polycythemia, one of the big issues is viscosity. If our blood is more viscous, it doesn't move as easily. If the blood doesn't move as easily, the heart has to pump harder. It has to squeeze the blood harder, and that's going to cause our blood pressure to be elevated and have excessive blood pressure. We also could have excessive clotting in our bloodstream. If we have more solid components in our bloodstream, more red blood cells or erythrocytes, we're more likely to have a blood clot form or an embolism. And it could be a pulmonary embolism if that blood clot gets lodged in the lungs. Those aren't cool. Those can be very fatal. If that clot gets lodged in the heart, we could have a heart attack, or it get lodged in the brain, the clot becomes a stroke. So generally speaking, having excessive polycythemia is not a good thing. We also could have not enough blood or red blood cells. That's going to be known as anemia. So when we don't have enough erythropoiesis, when we're not making enough red blood cells, we have anemia. Usually it's due to inadequate erythropoiesis. A secondary cause could be inadequate hemoglobin synthesis. If you don't have the hemoglobin, you can't make the red blood cell, or it's much more difficult to make a red blood cell. Symptoms that are caused from anemia include kidney failure, or you could have insufficient erythropoietin, so the kidney just doesn't make enough erythropoietin. We could also have dietary causes. My wife, for instance, has iron deficiencies. So every day she takes a little iron tablet, and if she skips her iron tablet for a couple weeks, she starts passing out randomly, and she'll just fall over. She's like a fainting goat. We could also have pernicious anemia. Pernicious anemia is when instead of not making enough hemoglobin so we can have enough red blood cells, we're instead going to destroy the red blood cells that we have. So we don't have the ability to get that vitamin B12 in. That's going to lead to destruction of red blood cells, or I should say elevated destruction relative to production. More are destroyed than are produced. So we have a net loss of red blood cells. We could have hypoplastic anemia, so that where we don't make enough red blood cells, we slow the process of erythropoiesis. Or we could have aplastic anemia. Aplastic anemia is when we stop making red blood cells altogether. Individuals that are undergoing bone marrow therapy and chemotherapy for blood cancers oftentimes are going to have the aplastic anemia because they have to have all of the red bone marrow cells destroyed. Will they reset those cell lines to hopefully remove all the cancer-forming cells? Question, comment? Ooh, great question. Sickle cell anemia. Didn't make it on the slide. Sickle cell anemia is a lack of red blood cells because the red blood cells are misshapen due to a protein irregularly crystallizing in its matrix. The hemoglobin will be prone to forming jagged crystals in the cytoplasm. Those jagged crystals in the cytoplasm will then make the red blood cell look like a sickle and then be destroyed. We can have anemia from bleeding too much or from destroying our red blood cells also. So when we think of not enough red blood cells, think about what happens to you when you don't have enough oxygen in your body. You feel lethargic, the ability to think is reduced. It just, things just generally don't work as well. So individuals that don't have enough oxygen are going to have the lethargy, the shortness of breath. When we think of organs that are highly ATP dependent, so nervous tissue needs a lot of ATP, our kidneys need a lot of ATP, and our heart needs a lot of ATP. These very ATP dependent organs depend on aerobic respiration to make lots of ATP. And if we don't have enough oxygen, we don't have enough ATP, and these are the organs that are most heavily affected by anemia. We also find that our blood viscosity is low, and having that low blood viscosity means that our heart has to pump a little bit faster to help compensate for the drop in our blood pressure. We also are going to find that we can have a reduction in blood osmolarity. The fewer blood cells that we have in our blood mean that blood osmolarity goes down. If blood osmolarity goes down, water will leave the bloodstream and hang out in the tissues. And we have a condition known as edema. So we have water or fluid retention in the tissues during this process because the osmolarity of the bloodstream has been reduced by the lack of erythrocytes. Let's go back to blood typing. When we think of blood types, there are gajillions of blood types. There's been over 200 different blood types identified within human beings, or 200 different surface marker proteins. But we don't care about most of them because for the overwhelming majority of those surface marker proteins, they're identical for all of us. And because they're identical from person to person, we don't necessarily care about them because they're not going to influence an immune response during a blood donation or blood transfusion scenario. There are, however, a couple proteins or glycoproteins that are going to influence our blood types that we do care about, that differ from person to person. And of those, we're going to talk about 3 very medically significant ones. There's some esoteric ones like the Bombay blood type. Read about those on your own outside of class. They're cool, but we're going to focus on the medically really big ones. Now, when we think of these surface markers, a generic name we can use for these surface markers is an antigen. And this is an immune term that, more broadly speaking, is any molecule that can trigger an immune response. Any molecule that can trigger an immune response is an antigen. And there are 3 antigens on our red blood cells that we really care about when determining blood types. As you can imagine, the structure of a cell is going to be influenced by the genetics of our cell. So what we find is that there's a strong genetic component to blood types. The blood type of your parents is probably going to be passed on to you. And we're not going to spend time covering dominant, recessive, and multiple trait characteristics or multiple allele characteristics at this point in time. We'll do that at the end of the semester when we get to hereditary function. There are genes and alleles that influence our blood types. Those blood types will have antigens or molecules on the surface of the red blood cell that trigger the immune response. We can also have an agglutinogen And these are going to be the I'm pretty sure I butchered that word, the agglutinogen, there we go. The agglutinogens are the specific antigens on the surface of the red blood cell that determine our blood types, that we care about for our ABO and rhesus factor blood typings. We also have antibodies. An antibody is going to be a protein that binds to the antigen. An antibody is an immune protein, sometimes referred to as a gamma globulin, sometimes referred to as an immunoglobulin or immunoglobulin. These are proteins that are made by a plasma cell, which we will talk about with the immune system type, a special kind of lymphocyte, the antibodies, and they are going to bind to those specific molecules that determine our blood type. They're going to bind to the agglutinogens. The specific antibodies that we care about for blood typing, the specific antibodies that bind to the agglutinogens, we call the agglutinins. So an agglutinin is an antibody that binds to a glutinogen. Or more broadly speaking, it's an antibody that binds to one of those surface markers on the blood cell that we care about. When we have an immune response with our red blood cells and we have an inappropriate matching, where we give the wrong blood type to a patient, we have the wrong kind of a transfusion, we can have lots of out-of-control clumping. The recipient's antibodies can bind to the donor's red blood cells and cause those red blood cells to clump together. And if we have lots and lots of clumping of the red blood cells, those red blood cells can't fit the blood capillaries. And we have microscopic blood clots forming in thousands of different places all over the body. Generally speaking, it's not good for the patient. You can have just a little bit of this happen, and the patient will have really bad symptoms. But if too much inappropriate blood is transferred, the patient will almost always perish. There's a name for this phenomenon known as disseminated intravascular coagulation, or DIC, D-I-C for short. Hopefully, you never interact with somebody who has experienced that. When we think of red blood cells, the red blood cell antigens we care the most about are the A, B, and rhesus factor antigens. Another name for the rhesus factor antigen, and rhesus is abbreviated with an RH. It's named after the rhesus molecule that the protein is isolated from. The single letter abbreviation for the rhesus factor is the D abbreviation. So we have the A, B, and rhesus factor, the A, B, and D antigens on the surface of the red blood cells. These antigens are made of glycolipids. So we have a carbohydrate attached to a lipid group, or more specifically, a carbohydrate attached to a phospholipid in the cell membrane. Those antibodies that we refer to as the agglutinins that bind to those very specific lipids are going to be found in the blood plasma. The antibody that binds to the A antigen we call anti-A. Anti-B binds to the B antigen, and anti-RH binds to the RH antigen. So if we're looking at our different blood types, and you're looking at a patient that has type A blood, that patient is going to have lots of A antigens on the surface of their blood cells. If that patient has type B blood, they'll have the B antigen on the surface of the blood cells. If they have AB blood, they have two different antigens. They'll have the A and the B antigens present on the surface of the red blood cells. The absence of A and the absence of B is referred to as type O blood. So type O blood, because you're probably wondering about type O in the back of your mind as we're talking about this. If somebody has type O blood, the absence of the A antigen and the absence of the B antigen. The reason we have a dividing line right here is because a different gene controls the rhesus antigen. If the rhesus antigen is present, that patient has a positive blood type. So they'll be Rh positive. So if somebody had the green, the blue, and the yellow antigens on the surface of the red blood cells, they would be AB positive because they would have all three of those antigens present. If somebody has the absence of A, the absence of B, and the absence of the rhesus factor protein, that individual will have O negative blood. When somebody is Rh negative, they have the negative blood type and do not have the rhesus antigen on the surface of their blood cells. So I was getting a little bit ahead of myself here. I pretty much said everything on the slide already. Type A blood has A antigens, B has B antigens, AB has both A and B, O has neither. And then when we think of the rhesus factor, the presence of the rhesus factor antigen gives you a positive blood type, and the absence of it gives you a negative blood type. This is kind of counterintuitive, but worldwide, the most common blood type is O. A lot of you in here are pre-nursing majors, and when you go into your nursing studies, you study different blood types based on countries. And you find that from one region of the world to another region of the world, different kinds of blood types are more common than other kinds of blood types. But worldwide, type O is the most common. So let's talk about blood typing and blood transfusions. If you take type A blood and you give it to a person who does not have type A blood, you're going to say, hey, wait a second, this does not belong in my body. There's this weird surface marker, glycoprotein or glycolipid, and this doesn't look right. I'm going to initiate an autoimmune response because this cell is a foreign invader cell. And they start to have their immunoglobulins, or the recipient's antibodies, start to attack and bind to the donated red blood cells. There's a common theme with blood transfusions. I'm going to repeat this over and over again. As long as you do not introduce anything new to the immune system will freak out when you give them something new and unexpected. So if I have type O negative blood, my blood cells will not have an A antigen, I won't have the B antigen, and I will not have the rhesus factor antigen. And if you donate A negative blood to me, so if I had the recipient, I was the recipient, and someone donated that A negative blood to me, you would be introducing something new to my bloodstream. You would be introducing an A antigen, and my immune system would trigger a response. Now, if we take type A blood right here and expose it to anti-A or type A antibodies, we'll have the clumping occur. And those red blood cells get clumped together and form little, not clots, but little clusters of cells. If you take type A blood and expose it to the B antibody, nothing happens, because the B antibody does not bind to the A antigen. If you take type B blood and expose it to the A antibody, nothing happens. But type B blood, when exposed to the B antibody, will have the clumping occur. And if you take type AB blood, it will clump in the presence of A antibodies and B antibodies. And then it's cut off on the bottom of the screen, but we have type O blood on the bottom of the screen. So it's lacking the A antigen and the B antigen. And when exposed to those antibodies, nothing happens in both situations. So as we're looking at this process, there are lots of different antigens on the surface of those red blood cells that can trigger the agglutination or the clumping. And if we receive a mismatched transfusion, so we have the inappropriate blood type donated, those red blood cells will be clumped together and will start to block small blood vessels or blood capillaries. We'll have microclots forming all over the bodies. So when we think of our kidneys, which filter our blood, we have lots of tiny blood vessels in our kidneys. Those tiny blood vessels can be shut down or blocked, and we can have kidney or renal failure. And I think this will be our last slide for the day. So we're thinking of blood donation and blood transfusion. If you do not have any antigens on the surface of your red blood cells, if you don't have anything that can trigger an immune response, you are the universal donor. And that magic blood type is O negative. O negative people don't have A, B, or the rhesus factor. They can donate to anyone and not have an immune response in the recipient. If you have type AB positive blood, that AB positive blood means that you are naturally going to have the A antigen, the B antigen, and the rhesus antigen in your body, and you will not produce antibodies that bind to them. And you are going to be the universal recipient. You can receive blood from anyone. One last point I need to emphasize. When donating O blood, or donating blood in general, you want to donate the red blood cells from the donor. You do not want to donate the donor's antibodies. So somebody with O negative blood will have A antibodies and B antibodies in their blood serum. You don't want to donate those antibodies to the recipient. So you take the blood that's being donated, you spin it down and separate just the red blood cells out, dissolve them in saline, and then donate those to the recipient. And that's all I got for today.